So in the carbohydrates experiment, what we're going to be doing are 3 tests that take advantage of any differences in structure of the carbohydrates, as well as differences in what functional groups are present in each carbohydrate. And remember, carbohydrate is just another word for sugar, and there are many types of sugars that we're going to encounter in this class. So the 3 tests that we're going to be doing are tests using the Benedicts reagent, which has a blue color associated with it. We're also going to be doing a test using the Carbohydrates reagent, which also has a blue color. And then we're also going to be, the 3rd test we're going to be using involves the use of Celawanos reagent, which is a little bit more colorless. So when we add our different reagents to our different sugars, we're going to be making some qualitative observations. And what that means is we're going to see, we're going to try to record any color changes that occur. Keep in mind also that the lack of a color change is a perfectly valid observation, right? And that's actually very, very important for the experiment today. So in order to interpret those color changes, those qualitative observations, you would need to make sure that you read the experiment ahead of time so that you know what the color changes correspond to in terms of the identity of the carbohydrates. Okay, so we're going to be doing these 3 identification tests for carbohydrates. And just to remind you, the Benedicts test is a test that detects reducing sugars versus non-reducing sugars. The Carbohydrates test using the Carbohydrates reagent is able to distinguish monosaccharides versus bigger types of sugars. So monosaccharides have just 1 sugar unit, whereas di and tri and polysaccharides have multiple sugar units linked together. And then the 3rd test we're going to be doing is using the Celawanos reagent, which is able to distinguish between a ketose, which is a sugar that has a ketone group in it, versus an aldose, which is a sugar that has an aldehyde group in it. So I have my test tubes here labeled 1 through 12, just like your instructions say. And what I've done is I've separated them into groups of 4. Right? We have 3 groups of 4 that correspond to the 3 different tests that we're going to be doing. In each of the corresponding test tubes, I'm going to add 20 drops of each of the reagents. So in my first group of 4 over here, in test tube 1, I've already added 20 drops of my Benedicts reagent, and I'm going to repeat that for the other 3 test tubes in this pool. And then I'll add 20 drops of my Carbohydrates reagent into these test tubes 5 through 8, and then 20 drops of the Celawanos reagent in test tubes 9 through 12. Okay, so now that I have my reagents already set up, so I have my Benedicts reagent, I have my Carbohydrates reagent, and I have my Celawanos reagent, the next thing to do would be to add the actual sugar solutions to the corresponding test tube. So I have 4 different sugars that I'm going to be looking at today. I have 2% fructose. I also have 2% glucose. I have 2% sucrose, and I have 2% lactose. All right, so I know it may be difficult to see the names on these, but what you want to pay attention to is that all 4 of these sugar solutions are actually colorless. So what I'm going to do is, according to the system laid out in your procedure, I'm going to add, for example, 10 drops of the 2% glucose solution into test tubes 1, 5, and 9. So there's my 2% glucose. I'm going to take my dropper and I'm going to add 10 drops of the glucose solution to test tubes 1, 5, and 9. So here's test tube 1. I'm going to add 10 drops. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And when you add 2 things together in a test tube or in any container, it's a good idea to swirl as you mix it, or you can flick it at the base of the test tube to make sure that everything is mixed uniformly. So I'm going to repeat that process according to the instructions, and I'm going to add the corresponding sugar solution to the proper reagent. Okay, so I've added all of my sugar solutions to the corresponding reagents, and what you'll see, for example, is that the overall color doesn't change when I added the sugar solutions, because they were all colorless, right? We ended up with a bigger volume in each case. So these are my sugar solutions with the Benedicts reagent, sugar solutions with the Barford's reagent, and the sugar solutions with the Celawanos reagent. So what you probably would have noticed in the background is here I have my boiling water bath set up. So this is just a beaker with about 250 mils of tap water, and it has a nice rolling boil going on. So I'm next going to place test tubes 1 through 6 into my boiling water bath, and I'm going to let it sit there for about 5 minutes, and then I'll take a look. I'll remove it after 5 minutes and let it cool down and take a look at any color changes that have occurred. So after 5 minutes have elapsed, I've gone ahead and removed test tubes 1 through 6 from the boiling water bath using a test tube holder. And what I'm going to do next is put in the remaining 6 tubes, test tubes 7 through 12, into the boiling water bath as well for 5 minutes. Okay, so I went ahead and I removed the remaining 6 tubes from the boiling water bath, and I allowed them to cool down. And now we're going to take a look at what colors all 12 of these tubes actually turned into, right? And I would encourage you, as we go through all 12 of them, that you have your experiment handy so you can sort of match up the color changes to what they actually mean. All right, so this is test tube 1. Test tube 1 contained glucose in the Benedicts reagent. This is test tube 2. Test tube 2 was fructose in the Benedicts reagent. This is test tube 3, which had sucrose in the Benedicts reagent. And this is test tube 4, which contained lactose in the Benedicts reagent, right? So remember, the Benedicts reagent is a test for reducing versus non-reducing sugars. Test tube 5, this is the Barford's test with glucose, right? If I shake this up here to sort of get all the solid particulates distributed evenly, you can see the color. It's a really dark, I know it's kind of difficult to see, but it's a really dark red color associated with it. Same thing for test tube 6, right? All the solid here has fallen to the bottom. If I shake it up and agitate it, right, you can see it's got a nice dark red color associated with it. Test tube 7 is sucrose in Barford's reagent. Test tube 8 is lactose in Barford's reagent. Test tube 9 is glucose in Celawanos reagent. All right, and the reason why I put the white paper as a background is so you can see sort of subtle color changes. That's test tube 9 is glucose in Celawanos reagent. Test tube 10 is fructose in Celawanos reagent. Test tube 11 is sucrose in Celawanos reagent. And then test tube 12 is lactose in Celawanos reagent. And there you have it. Those are the 4 different sugars: glucose, fructose, sucrose, and lactose, and how they behave with these 3 different tests: the Benedicts test, the Barford's test, and the Celawanos test, we can use to identify what sort of functional groups and by extension what types of structures we have in those compounds.